Hello everyone. In this video, we are going to be talking about the nervous system. So the nervous system is the part of our body that controls our thoughts, feelings, and starts with actions of the nervous system, which we're going to get into. So some neuroscience concepts. So there's something called neuroplasticity, and this is the brain's physical ability to change and develop throughout the lifespan. So these include things like neuronal pathways, and throughout life, we can actually increase these neural pathways, which is why like habits and things we can reinforce because of that neuroplasticity. The brain is able to change. Adult neurogenesis, this is when new neurons can be generated in adults. So we were just kind of talking about that with habits and things. Experiences and genetics influence brain development. There's also something called synaptic pruning. This happens in adolescence, and it's when the synapses that are relevant persist, refine the efficiency of the brain. So they kind of prune those synapses in order to make the brain run better. But this is interesting when it comes to mental illness, because sometimes this synaptic pruning can be part of the problem that then leads to these brain disorders. So some things to note: chronic stress and chronic substance use disorder can negatively affect neuronal plasticity. Psychotherapy, psychopharmacology, enjoyable activities, exercise, and meditation can produce positive changes on brain neural functioning. Synaptic or neurotransmission and neurotransmitters. So this is the transfer of information from one neuron to another. We're going to talk more about that. It's stimulated by what's called an action potential in the axon, where then neurotransmitters are released by the presynaptic neuron into the synapse. So this is just kind of an overview of what we're going to be talking about. I know it's a lot of words, but it's a good way to preface what we're going to discuss later. When action potentials stimulate the axon, the neurotransmitters are released from the presynaptic neuron into the synapse. So presynaptic is like where the neurotransmitters come from. The synapse is the space between two neurons. And when these neurotransmitters are in that space, it activates the receptors on the postsynaptic neurons. That's the one like accepting those neurotransmitters. When the channel is open, ions exchange and impulse cause reaction in the receptor cell. Receptor channels can close and the NT reuptake to the so the neurotransmitter reuptake into the presynaptic membrane occurs. So we're going to talk about this more, but you have your presynaptic cell or neuron that is releasing those neurotransmitters into the synapse, the space between. Then a postsynaptic neuron that is sucking up those neurotransmitters. However, when all the receptors, once they're on there, maybe they're not taking up anymore, and now you have excess neurotransmitters in that synapse. Well, what's going to happen is that presynaptic cell, the one that released them in the first place, is actually going to take them back up into it. And this is called the reuptake. When we talk about things like SSRI, so serotonin, reuptake inhibitors, what that is doing is actually blocking that reuptake in the presynaptic neuron. So it's leaving more serotonin in the synapse. So just interesting. You're going to hear these things as we continue. The second messengers within the neuron continue neurotransmission through chemical and electrical processes. And just some notes: the work of the brain is at the synapse. Healthy lifestyle maintains optimal functioning of synapses, and there are more than 50 neurotransmitters in the brain. And then neurodevelopmental basis of psychiatric diseases. So conditions that originate during brain development include things like schizophrenia, depression, autism, and ADHD. Conditions, so that's just something to note. So the nervous system. So you can see these pictures at the top of the page. So we just kind of talked about let's talk about the green one first, because we just talked about that. So you can see the presynaptic neuron is up here, and then you can see these little blue neurotransmitters, and then we can see, so normally it's coming this way, and those neurotransmitters are released into the synapse. That is this space here. And then down here, this is our postsynaptic neuron. And you can see that that is taking up the neurotransmitters. Then we also have here, we have a neuron and it's showing the direction of the action potential. So we can see on one side of the neuron, we have the dendrites. Those are like long, it's kind of like a cell body and then little things that hang off of it. And then a cell membrane. And then you go along this axon, you can see those myelin sheaths. That's that kind of green that is on there. When you have illnesses like MS, it actually breaks down those myelin sheaths. But if you have a normal one, that is what it looks like. And then our action potential is moving from the dendrites in the cell membrane down to the synapse at the other end, which is then connecting to the dendrite of another neuron. So nervous system. So again, this is going to control our senses, how we interpret things, and issues commands as a response to the environment. For example, I touch something hot and my brain says, oh my God, it's hot, and I pull my hand away, right? So because of the environment, I'm getting a command, and it is really the microprocessor of the brain. When we talk about neurons, these are basic nerve cells that conduct information electrically along incoming axon fibers. So again, that is this kind of long thing covered in the green. This is a long extension of the nerve cell body, and it sends impulses and outgoing dendrites, a shorter branch extension that receives the stimuli. So the dendrites receive the stimuli, and we can see that here as well. The glial cells are structures that form the myelin sheaths around the axon and provide protection and support. When a neuron is stimulated past a necessary threshold, messages are sent across the plasma membrane of the neuron through a process called action potential. And communication between axon terminals and neurons is done chemically through neurotransmitters that are released into the synapse or junctions between the neurons, which we talked about. When it comes to neurotransmitters, information is conveyed along neurons both electrically and chemically. And when we say chemically, those are the neurotransmitters. Synapses transmit information using chemicals synthesized from dietary substrates called neurotransmitters. So, classification requirements for neurotransmitters. So they must be present in the nerve cell terminal. Stimulations of the neuron must cause the release of neurotransmitters in sufficient quantities to cause an action to occur at the postsynaptic membrane. Effects of exogenous transmitters on postsynaptic membrane must be similar to those caused by stimulation of a presynaptic neuron. A mechanism for inactivation or metabolism of a neurotransmitter must exist in the area of the synapse, and exogenous drugs should alter the dose-response curve of the neurotransmitter in a manner that is similar to naturally occurring in the synaptic potential. So when we say exogenous, that's just like if you added a medication that was giving a specific neurotransmitter, something that's not necessarily coming from the body, but coming from somewhere else. The neurotransmitter will be released from the presynaptic neuron across the synapse that binds to a specific receptor on the postsynaptic neuron, and specific criteria must be met for a molecule to be classified as a neurotransmitter. Categories of neurotransmitters include things such as monoamines, amino acids, cholinergics, neuropeptides, and monoamines or biogenetic amines such as dopamine, norepinephrine, epinephrine, serotonin. And these are kind of like the big ones when it comes to psychiatric medications. These stimulations occur in a sequence from the stimulation point of one neuron to its contact with another neuron. So talking about some of those neurotransmitters we just discussed. So dopamine, this is important to note for the exam. You need to know what part of the brain that the neurotransmitters are being created in, and what it is that those neurotransmitters are responsible for. So dopamine is produced in the substantia nigra and the ventral tegmental area. Precursor is tyrosine. It's removed from the synaptic cleft by monoamine oxidase enzymatic action. So there's 4 dopamine pathways. And again, you are going to have to know this for the exam. We're going to go over them later. So mesocortical, mesolimbic, nigrostriatal, and tuboinfundibular. And I have a very funny way that I memorize these that is semi-inappropriate, but it worked for me in order to memorize them. So sometimes you can take these words and kind of put together like a little story to help you memorize, because it's just, these are very complicated terms, right? So dopamine, when it is increased, we see things such as schizophrenia or psychosis. And when it is decreased, we see substance use anhedonia and Parkinson's disease. Then we have norepinephrine. This is produced in the locus ceruleus of the pons and medullary reticular formation. A precursor is tyrosine, and it's removed from the synaptic cleft and returned to a storage via an active reuptake process. Major neurotransmitter implication in mood, anxiety, and concentration disorders. When decreased, we see depression. When increased, we see anxiety. So for example, when I say, think about a funny story you could tell yourself. When I was trying to memorize where these neurotransmitters were made, and I just remember this one off the top of my head, norepinephrine, to me, that's like the fight or flight. And when I think fight or flight, I kind of think like war. And in my head, medullary reticular formation sounds military. It sounds like, because I don't know, medullary, like, I don't know. It just sounds like a medallion, like, and just so, and reticular, like, formation makes me think of like army people in like a certain formation. So I remembered that as like, you know, norepinephrine was medullary reticular formation, thinking of like war. And then like the pawns also kind of sound like it would be war, because you're like pawning and trying to, you know, get your opponent. So that is like a silly story that helps you remember these things with these super difficult terms. And then I'm trying to think for dopamine, the way I remembered it. So see, the one that comes to my mind is semi-inappropriate, so I don't want to go too inappropriate with this, but when I think dopamine is like happy, when you're doing, you know, things that might make you happy. When I think of substantia nigra, for some reason, it makes me think of like Niagara Falls, and vagal tegmental area, kind of makes me think of like a triangle for some reason. I don't know if anyone else, if this makes you think of it, because it's like tegmental area. So I think of the Bermuda Triangle. So when I think of Niagara Falls and the Bermuda Triangle, I think of like travel, and travel makes me happy, which is dopamine. So again, kind of like a silly story, but to be able to memorize these things. And then, so epinephrine, also known as catecholamine, is produced by the adrenal glands. Epinephrine system also refers to the adrenergic system. So the way I remembered this is, Our adrenal glands, when we get stressed, they release stress hormones, which aren't good for us, which makes me think of epinephrine, because we don't want to be in that like fight or flight all the time. So that's just how I remembered that one. All right. So serotonin, this is produced in the raphe nuclei of the brainstem. Precursor is tryptophan, removed from the synaptic cleft and returned to storage via an active reuptake process. Remember, we talked about the presynaptic cell will reuptake it back into it. Major neurotransmitter implicated in mood and anxiety disorders. So if it's decreased, we see depression, OCD, anxiety, and schizophrenia. Cholinergic acetylcholine. This is synthesized by the basal nucleus of NERT. Precursor are acetylcholine enzyme A and choline. So how I remembered this one is, for some reason, basal nucleus of NERT made me think of like aliens. Like I just think of basal nuclei of NERT. It sounds like some planet in outer space. And then I remembered like when people take cholinergics, I mean, technically they take anticholinergics, but when people take anticholinergics, they can get anticholinergic toxidrome syndrome, which makes them like a little crazy. So that's how I remembered that. Kind of crazy thinking aliens. Again, you can make silly stories. It really helps, especially with this chapter, because there's just a lot of terminology that can get super confusing. So although my stories might sound ridiculous to you, make up your own kind of story, right? Write down cholinergic, write down basal nucleus of NERT, and then write a line and write what it reminds you of, and then create a story to tie those things together. So, when this is decreased, we see Alzheimer's disease, impaired memory, and increased Parkinson's symptoms. There's also neuropeptides like non-opioid type, substance P, somatostatin, opioid types such as endorphins. You don't need to worry about these ones. You're really not going to see these on the exam. And again, these are going to modulate pain and will be decreased in substance use disorder. There is a component with substance use disorder, specifically people who do take opioids, that they have a lower pain tolerance than people who don't use those substances. So it probably has something to do with this. So just an interesting fact. Increased levels of corticotropin releasing hormone in the amygdala, hippocampus, and locus coeruleus increases symptoms of anxiety. So just important to note. So then amino acids, we have glutamate, aspartate, amino butyric acid, also known as GABA. We have glycine as well. Glutamate is the most abundant excitatory neurotransmitter in the brain. Increased levels of glutamate will increase anxiety levels. It is a major neurotransmitter involved in the process of kindling, which is implicated in seizure disorders and possibly bipolar disorder. An imbalance implicated in mood disorders and schizophrenia. So if we have increased GABA, it can lead to bipolar and psychosis. Decreased can lead to memory and learning difficulties, and also can play a role with the negative symptoms of schizophrenia. We also have aspartate, which is another excitatory neurotransmitter. It works with glutamate. And GABA is the most abundant inhibitory neurotransmitter. The site of action of benzodiazepines is on GABA. Alcohol works on GABA, barbiturates, and other central nervous system depressants. When we have an increased amount of GABA, when we have a decreased amount of GABA, we're going to have anxiety, because GABA is calming. So decreasing it would result in anxiety. And benzodiazepines are used to bind with GABA receptors to potentiate anxiolytic or calming effects of GABA. Then we have glycine, which is another inhibitory neurotransmitter that works with GABA. All right, so we kind of went over all of the neurotransmitters. This is just a nice table to put it all together, all of the things we just talked about. When you see the word agonist, as you can see at the top, you'll see agonist. What that means is when we increase it. And then when you see antagonist, that means when it's blocked. So for example, dopamine, when we take antipsychotics, we are blocking dopamine. So that is an antagonist. However, when we take things like cocaine or amphetamines, ADHD medication, Wellbutrin, we are actually increasing dopamine. So that would be an agonist. It's also going to go over the general role and function, which we kind of just talked about, and then the symptoms that you will see if it's in a deficit or excessive. It also talks about the receptors. You don't need to worry too much about this. I mean, I would definitely know that serotonin is the 5-HT, and then there's a bunch of different ones, right? Dopamine's obviously D, GABA is obviously GABA. Know that opioids work on the mu. But besides that, you don't really need to memorize all of those. This is like a good sheet that you could study from if you need to too. All right, so neurotransmitters. So recovery and degeneration of neurotransmitters. So after a neurotransmitter reaches the postsynaptic neuron, it may then diffuse off its receptor to be destroyed by enzymes or to be transported back to the presynaptic neuron for reuse. Enzymatic destruction occurs either in the cytosol or in the synapse. The neurotransmitter can be destroyed by the enzyme monoamine oxidase, MAO, which is why an MAOI is a monoamine oxidase inhibitor. So we are stopping this enzyme from breaking down and destroying neurotransmitters. The problem with those medications is there is a issue with eating certain foods that causes a tyramine buildup. We'll go over this when we get to that lecture. And it's just a really tricky medication for people, so not used super often. You pretty much can't eat many yummy things, so it's tough. All right. So reuptake pumps, we kind of talked about these, right? You have your presynaptic cell or neuron, your postsynaptic neuron, it's released that neurotransmitter in it, and then the reuptake pump is what is on the presynaptic cell that pulls those neurotransmitters back up into the cell in order to be recycled. And again, we can see that over on the side. So at the point of contact called a chemical synapse, a substance is released that stimulates or inhibits the action of the adjoining cell. This network fans out across the body and forms the framework of the nervous system. Synapses or synaptic clefts are the connection site and the area of communication between the neurons and the neurotransmitters. The synapse converts an electrical signal from the presynaptic neuron into a chemical signal that is transferred to the postsynaptic neuron. So again, starts as electrical, then goes to chemical. These neurotransmitters are released in the synaptic cleft, and the two phases of an action potential are depolarization. So this is the initial phase of action potential when sodium and calcium ions flow into the cell, and then repolarization, which is the restoration phase when potassium leaves the cell or chloride enters the cell. So those are the two phases of an action potential. So problems in either the structure or chemistry of the synapse interrupts the normal flow of impulses and stimuli, which then contributes to symptoms commonly seen in psychiatric disorders. So there are types of neurons. There are three general types. So we have sensory neurons. These transmit signals to the central nervous system from the sensory receptors associated with touch, pain, temperature, hearing, sight, smell, and taste. Again, our senses. Then we have motor neurons. So these are going to transmit signals from our central nervous system to the rest of the body as by signaling muscles or glands to respond. And then we have interneurons or relay neurons that signal between neurons. A neuron has three basic parts. We kind of talked about this, right? We have our cell body, also called the soma. It is where it has a cytoplasm with a cell membrane and then has those dendrites coming off of it. We have that axon that transmits the impulses, and it is insulated by the myelin sheaths with gaps known as nodes of Ranvier. And the myelin sheaths insulate the neurons, and it terminates at the synapse, and then dendrites are what receive those impulses from the sensory receptors or interneurons and transmit them towards the cell body. Really, their main function is to receive incoming signals. All right, so central nervous system. Our central nervous system is in the center, right? So we have our brain, we have our spinal cord. It controls our thoughts and muscle movements. We have efferent nerves. These are motor, which relay commands from the central nervous system to the body. And then we have afferent nerves. These are our sensory, and they relay sensory information from the body to the central nervous system. The spinal cord is encased in a bony structure of vertebrae, which protect it. Its nervous tissue functions include mainly limb movement and internal organ activity, and major nerve tracts ascend and descend from the spinal cord to the brain. So within the nervous system, we have various parts. So you can see we have that central nervous system that we just talked about. And then we have the peripheral nervous system, which we're going to talk about. Within the peripheral nervous system, we have the autonomic nervous system, the somatic nervous system, and the visceral nervous system, which we're going to talk about. And within the autonomic nervous system, we have the sympathetic, parasympathetic, and enteric nervous system. So first, we're going to talk about the somatic nervous system. So these are our five senses and the voluntary movement of skeletal muscles. Efferent muscles bring signals from the central nervous system to organs and the muscles, and then afferent bring the signals from the sensory organs system to the muscles to the central nervous system. So efferent is going from our central nervous system out, and afferent is going out in. So involuntary movement. So within the somatic nervous system, there are some involuntary movements. It is not all, it is only reflexes. So really this only controls our reflexes. And the somatic nervous system mostly controls our voluntary movements, which is controlled by acetylcholine, which causes our muscles to contract. All right, then we have the visceral nervous system. So this controls the nerves that relay information between the central nervous system and our visceral organs. And then we have the autonomic nervous system. This is going to be what has our sympathetic, parasympathetic, and enteric system. So it regulates internal body function. It's responsible for most of our involuntary movement besides our reflexes. It has our sympathetic, parasympathetic, enteric. It is composed of peripheral nerves that connect to the central nervous system, muscles, and glands. And it includes cranial nerves just outside the brainstem, which we're going to talk about. So these are the 12 cranial nerves. You will see this on the exam. However, I was surprised when this potentially was seen on an exam. They mentioned which nerve it was, which made me feel like, why did I memorize all of these things? However, it is a concept we are being tested on, so try your best to remember them. Just because that was the way it was presented to me doesn't mean that's going to be the way it presented to you. Here's how I remember the 12 cranial nerves. So we can see, I wanted to see, yeah, okay, this is a better way. So these are kind of the 12 cranial nerves in a pretty picture format, but for how we're going to memorize it, let's look at it this way. So you can see the nerve number here, and then it kind of comes over this way. So olfactory, optic, ocular motor, trochlear, trigeminal, abducens, facial, vestibulocochlear, glosseopharyngeal, vagus, accessory, hypoglossal. So a lot of words, right? Here's how I remember it. So on, they traveled and found Voldemort guarding very ancient Horcruxes. I love Harry Potter, so this was an easier way for me to remember them. So if you can remember that, then you have all of your letters. So, when you sit down to take your exam, you're going to have a whiteboard or a piece of paper, and this should be one of the first things you write so that you will not have to forget it. And when you're in the stress of the exam, you're not freaking out. So, all right, so now you have all your letters. So number 1, we know is an O. There's a couple O's though. So here's how we remember it. Olfactory is 1. You have one nose. Your one nose is olfactory. It's smell. Optic. You have 2 eyes. So your second is optic. And then you remember that your last O is ocular motor. So we remember 2 optic for the eyes, but then our eyes also move. So ocular motor is 3. Okay, so same with the T's, right? We have 2 T's. So the way we can remember this is we have trochlear, and this is motor to the eye. So this one is looking down. So again, we're at the ocular motor is 3, our eyes are moving. And when we look down, that's 4. Okay, so then we have one more T, which is 5. We remember that's trigeminal. Trigeminal is the movement of the face. So having people clench their jaw, move their face around. So like we have the eye movements for 3 and 4, and then 5 is going to be like clenching the jaw. But again, it's your last T, trigeminal. And then abductans, this is... we have 2 A's. So you just have to remember like which one comes first. Again, abductans is movement of the eyes. So we're still movement of the eyes. So that one's kind of staying first. So that is going to be our 6, being able to look side to side. And then F is facial. There's only one F. So now we're on to V, and we do have 2 V's. So how do we remember which one comes first? So the vestibular cochlear comes first, and this is sensory to the ear for hearing and balance. And then we have vagus, which is the pulse. So the way I remember this is we're sticking with the face first. Like we're still up here. We're clenching the jaw. We're moving our eyes. We're smiling, we're staying up here. So then we're going to the ears, having them close their eyes, snap their fingers. Can they hear? So that is the first V. G, there's only one G, so that makes it easy. Glosseopharyngeal is having them swallow. And now we're on to our second V, which is the vagus nerve. So we can check a pulse for that. But again, it's our last V. And then we have accessory, which is muscles of the neck. So again, we're moving down from the face. So now they're swallowing. Now we're checking their shoulders. And then the last one is H, which is hypoglossal, which is sticking out their tongue. So I always think like at the end, we're done, we're sticking out our tongue. Although there were quite a few different things there, but I always think of like, okay, we got to the end. So you just stick out your tongue, make a funny face. Okay. All right. But again, there might be other ways that you remember this, but that is kind of the way my brain goes through it. All right. So the peripheral nervous system. So now we're moving from the central in the middle to the peripheral. So now we're on the outside. So we have our parasympathetic and our sympathetic. So parasympathetic is really our rest and digest. It is what restores the body to a state of calm. Truly, 90% of the time, this is where we want to live, although we don't. And then the sympathetic is that fight or flight. When we see, when we have a seen or perceived threat, that kind of joust of energy, heart rate increase, sweating, And then enteric is located in the GI tract. It connects the brain and the digestive system through nerves. It controls secretion, blood flow, hormone release, motility, and the movement of food through the digestive tract and the digestive process. When one is activated, the other one in ways is deactivated. So for example, if you are in a parasympathetic, your digestive system is digesting food, it's pushing it through, it's having secretions. Whereas if you're in a sympathetic, then that's actually inhibited. It's the opposite. Things aren't going to really be moving. And same kind of thing, parasympathetic, it's going to lead to contractions of the bladder, whereas sympathetic is going to relax the bladder. That's why when people get really scared, they might urinate themselves. It's just part of that sympathetic response. When we have a sympathetic response, we have dilated pupils. The way I like to think about this is like the pupils are opening to kind of take everything in, although it works the opposite way, which is interesting. When you're in the sympathetic, you actually have tunnel vision. But I just think of to remember that they dilate, it's like, oh God, something's happening, right? And everything's just like dilate. The eyes are dilating, although everything else is okay. So that was kind of a big overview of the nervous system. Now we're going to talk about neuroanatomy and the brain. You will need to know this for the exam. We're going to talk about brain tissue, the areas of the brain, what they do, things like that. Brain tissue is categorized as either white matter or gray matter. Just FYI. I don't know if that will be on the test, but white matter is the myelinated axons of neurons, where gray matter is composed of nerve cell bodies and dendrites. It is the working of the area of the brain and contains synapses, the area of neuronal connection. The outermost surface of the brain is structured to contain what are called grooves and dips of corrugated wrinkles within the brain tissue that provide anatomical landmarks and references. These have names. So sulci are small, shallow grooves, and fissures are deeper grooves extending into the brain. Gyri are raised tissue areas. And distinct anatomical areas of the brain, which we're going to go through. So this again is a chart. I feel like when trying to remember things, charts are nice. So there's two kinds of areas of the brain we're going to talk about. So we have the brainstem, which consists of the pons, medulla oblongata, and the midbrain. So that is the bottom piece of our brain. It's like the stem that's kind of holding the brain up, but that's not really holding it up, but it kind of looks like that, right? If you saw a picture. And this regulates blood pressure, respirations, level of arousal, and digestion. That's why when some people get an injury to the neck, high up, it can actually stop, because they're injuring their brain stem, they can just stop breathing and be dead, because that's what that area regulates. If it is damaged, we can see things like anxiety, depression, and personality changes. Specific conditions plays a role in PTSD, paralysis, psychosis, coma, and death. And then the cerebellum, this is the brain itself. It coordinates balance, posture, movement, memory, impulse control, cognition, and language. Damage can lead to ataxia, tremors, emotional blunting, and lack of inhibition. And this can be seen in autism, ADHD, depression, bipolar disorder, schizophrenia, can be things that come from this area. All right, so let's talk some more. So the cerebellum, again, this is like the entire brain itself, not the brainstem. It's the larger part of the brain, which is divided into two halves. We have the left and the right brain. Normal functions require effective coordination of the two hemispheres. The cerebrum includes the cerebral cortex, the limbic system, the thalamus, hypothalamus, and basal ganglia. On the left hemisphere, this is dominant in most people and controls most right-handed body functions. Or right-sided, sorry, if you write with your right hand, you're probably left hemisphere dominant, but it controls most of your right body functions. And then the right hemisphere controls most of the left body functions. Normal functioning requires the coordination of these two hemispheres. Both hemispheres are connected by a large bundle of white matter called the corpus callosum, an area of sensory motor information exchange between the two hemispheres. And each hemisphere is divided into 4 major lobes, which work in an interactive and integrative manner, and each with a distinct function. You are going to need to know what these functions are. We're going to talk about them. So first we have the frontal lobe. The frontal lobe is really that front part of our brain. So we can see it here. This is the front part of our brain. This is the back part. This whole thing is the cerebrum. And this is the brain stem down here. You can see it better here. All right, so the frontal lobe, we are here. Something important to note about the frontal lobe is this is also where Broca's area is. And this is expressive speech. So if somebody has injury to Broca's area in the frontal lobe, they will not be able to express their needs because they will have difficulty with expressive speech. They will be able to understand what you're saying, but they will not be able to express what they feel. So the frontal lobe controls the planning, reasoning, imagining area of the brain. This is where executive function and decision-making largely occur. It's responsible for judgment and short-term and working memory. It plays a role with motor function. It's responsible for controlling voluntary motor activity of specific muscles. It has a premotor area that coordinates movement of multiple muscles. It has an association cortex, which allows the multimodal sensory input to trigger memory and lead to decision-making. It really is the seat of executive functioning. It has working memory, reasoning, planning, prioritizing, sequencing behavior, insight, flexibility, judgment, impulse control, behavioral cueing, intelligence, and abstract. Big thing for the exam. It plays a role in our personality. So people who have injury to their frontal lobe can have significant personality changes. I've seen this in practice. People get in a bad car accident or something like that, injury to that frontal lobe, and their family just doesn't even recognize them anymore as far as their personality. And it can also lead to changes with emotions and intellectual changes as well. Injury to that area. All right, so that was the frontal lobe. Now we're going to talk about the temporal lobe. So you can think about the temporal lobe. We can see it here. It's on the side, but think of your temples, right? The temporal lobe is on the side there next to the temples. This area controls speech. It is located at the left and right side of the brain. It's responsible for all auditory input, processing, and output. Now, when we say controls speech, the area of this brain, the area within the brain that's important to note in the temporal lobe is called Wernicke's area. So we were talked about in the frontal lobe, we have Broca's, which if there's damage to that, then you have difficulty expressing speech. However, in the temporal area, we have a Wernicke's area. And if there's injury to this, we have difficulty with receptive speech or language comprehension. So somebody that has injury to this area will not understand what you're saying, but yet they'll still be able to speak. So they might be speaking nonsense. They might not be able to respond to what you're saying, although can express other things. So just take note of that. This is the primary auditory area. Plays a role with memory, emotion, integration of vision with sensory information, and problems with the temporal lobe can lead to visual or auditory hallucinations. Big star. This may or may not be on the exam, but very important. There are other areas of the brain that can lead to visual and auditory hallucinations. However, there probably won't be it usually has to do with like too much dopamine in certain areas. But just to note, if there's a question that's like what part of the brain, injury to what part of the brain might cause visual or auditory hallucinations or problems in what area, it's going to be the temporal lobe. It can also lead to aphasia and amnesia. Then we have the occipital lobe. So the occipital lobe is in the back of the head. We can see it here. It's in the green. And this controls visual information. Nerves from the eyes enter directly into this lobe. So you think our eyes are here, the nerves go straight back into that lobe. It's the primary visual cortex, has an integration area that integrates vision with other sensory information, and problems with this area can lead to visual field defects, blindness, and visual hallucinations. Then we have the parietal lobe. This is located slightly towards the back of the brain and the top of the head. So the parietal is kind of like right here. It's behind, the frontal lobe, it's behind it, but it's above the occipital. You can see it over here in the light purple. It's responsible for sensory input as well as spatial positioning of the body. It has a primary sensory area, plays a role in taste, reading, and writing, and problems with this can lead to sensory perceptual disturbances and agnosia. So the cerebrum, cerebral cortex, controls behaviors, speech, cognition, judgment, perception, and motor function. It controls the opposite side of the body. So we talked about this when we say cerebral cortex, that is the whole brain, not counting the brainstem. We kind of talked about left controls the left controls the right side of your body. The right side of the brain controls the left side of the body. Sensory information is relayed from the thalamus and then processed and integrated into the cortex. Okay, so like I said before, I really like charts in putting things together. So with this, this is everything we just talked about in a chart form. So again, the frontal lobe controls, executive functioning, personality. Damage is going to result in this. Specific site conditions are this. So again, if you want to print this out to be able to kind of go through, but we really just went through all of this information. So now let's talk about the limbic system. So this is the regulation and modulation of emotions and memory. It's composed of the hypothalamus, the thalamus, hippocampus, and amygdala. This is kind of like in the center of your brain. Let's see, do I have a picture? Yeah, here we go. So you can see, it's right. So we can see like the hippocampus, the amygdala is here. We can see our pituitary, the hypothalamus is here. So it's kind of like in the center, kind of at the top of the brainstem. All right. So the hypothalamus plays a role in regulatory functions such as appetite, sensation of hunger, thirst, water balance, circadian rhythms, body temperature, libido, and hormonal regulation. These, again, you need to know these for the exam. The thalamus is sensory relay station, except for smell. So it modulates the flow of sensory information, regulates emotion, memory, and affective behaviors. The hippocampus regulates memory and converts short-term memory into long-term memory. The amygdala is responsible for mediating mood, fear, emotion, and aggression. It connects sensory smell information with emotion and processes memory and emotional responses. For the exam, the amygdala is very important. They're all important to remember, but the amygdala with the sense of smell and remembering information and past experiences related with emotion. So trauma, typically if there's a question that has to do with like trauma, PTSD, flashback, anything like that, typically the issue is with the amygdala. Okay, so again, charts. Charts are nice. So we talked about the amygdala, regulates basic powerful emotions, fear, rage, and sexual desires. Effects of damage can be irritability, anger, aggression, in specific conditions, PTSD, panic disorder, autism, depression, schizophrenia. So again, this is just kind of an overview of what we just went over. Hippocampus is involved with memory and converting short-term memory into long-term. Again, these are things you're going to have to remember. Think of funny stories. So for me, hippocampus makes me think of a hippo. So then I kind of just thought of a hippo with a graduation cap on, and that it's involved with like memory and converting short-term memory into long-term memory and learning. So I don't know. That's how I remembered it. But again, these are all things you can come up with your own little stories. The thalamus relay station that connects the cortex to the limbic system affects mood, body movement, and strong emotions, impaired when people are overwhelmed with information. It plays a role in schizophrenia, PTSD, and memory problems. And then the hypothalamus, this is what maintains homeostasis and basic needs. So eating, drinking, temperature regulation, sleep-wake cycle. If it's damaged, we can have disturbed sleep, eating, changes in body temperature, and emotional instability. We can also see it with specific conditions such as depression, anorexia, and violence. So the basal ganglia, so we can see that the basal ganglia or the corpus striatum, if we look at this brain right here, we can see, hold on. The basal ganglia is the areas of red, and then related structures are the areas of blue. So the basal ganglia is a feedback system to modulate and stabilize somatic motor activity. It plays a role in movement initiation, complex motor functions in learning, and automatic actions such as walking or driving. It contains extrapyramidal motor systems on a nerve track. It functions in involuntary motor activity. Many psychotropic medications can affect the extrapyramidal motor nerve tract, causing involuntary movements. It contains both the caudate, which is innervated by dopamine neurons from the substantia nigra, integrates spatial information and working memory with motor behavior, limb movement, speed, accuracy, and posture. And the putamen, not sure how you say that, which is comprised of afferent sending pathways exerting an inhibitory effect on the thalamus. Problems with the basal ganglia can lead to bradykinesia, hyperkinesia, and dystonias. And then the brain stem. So now this is the stem at the bottom of the brain. This is made up of cells that produce neurotransmitters. It includes the midbrain, the pons, medulla, cerebellum, and reticular formation. It includes the midbrain, which houses the ventral tegmental area and substantia nigra, which are the areas of dopamine synthesis. The VTA is thought to be involved with various cognition and emotional processes, and the substantia nigra is the primary associated with movement. Pons houses the locus ceruleus. The medulla, together with the pons, contains the autonomic control center that regulates internal body functions. And the cerebellum is responsible for maintaining equilibrium. It acts as a gross motor control system. Each hemisphere of the cerebellum has, we already went over this, they control the opposite side of the body. Remembering cerebellum is the whole brain. Problem with the cerebellum can lead to ataxia, and a Romberg test is important for detecting deficiencies in cerebellar function. So Romberg test is when you have people hold out their arms and close their eyes and see if one drifts. The reticular formation system, this is the primitive brain that receives input from the cortex and supplies nerves to the thalamus, hypothalamus, and cortex. It regulates functions including involuntary movement, reflex muscle tone, vital sign control, blood pressure, respiratory rate, and is critical to the consciousness and ability to mentally focus, to be alert, pay attention to environmental stimuli. All right, this is important. This is a lot of words to know, okay? But we're going to do it. Okay, so the 4 dopamine pathways. So the mesolimbic pathway. So hyperactivity of dopamine in this pathway, the mesolimbic, mediates positive psychotic symptoms. Antagonism of D2, so when we decrease the dopamine receptors in this pathway, it will treat those positive symptoms. This is what we do with antipsychotics. It's reward oriented and it is associated with mood, psychosis, drug abuse, positive symptoms of schizophrenia. So the way So mesolimbic, we're going to go over the memorization trick last because I don't really have one for this one. So this is like the other one. All right. So mesocortical. So decreased dopamine in the mesocortical projection is postulated to be responsible for negative and depression symptoms of schizophrenia. So when I see mesocortical, I see the C. So the C makes me think cognition. So the cognition, planning, and behavior goes with the mesocortical pathway. And if what it's responsible for is cognition, when there's decreased dopamine, you're going to have negative and depressive symptoms because that cognition is not doing well. That's just how I remember that one. That's like the only like semi-appropriate. I feel like the other ways I remember this is just not, is like so strange. But okay, so, all right, so the next pathway, the nigrostriatal pathway. So for me, when I see the word nigrostriatal, I don't know why it makes me think of a dance. I don't know if it's because it looks like straddle in the middle and like I think like straddle and then I think of like dancing. So nigrostriatal makes me think of a dance move and it like makes me think of like the robot or something. So then I remember, oh, that's the one that is associated with tardive dyskinesia and like movements issues and EPS and dystonias and Parkinson's. So that's how I remember that one. You know, you just have to remember that it's really the blocking of D2 in this pathway that causes that. But I think of like antipsychotics block dopamine in the nigrostriatal pathway, and then that leads to the stiff movement. So we know dopamine's being reduced. So that's how I remember that one. I don't know if that will work for you guys, but that's what works for me. All right. This one is so inappropriate. It's so inappropriate, but sometimes I've been like mortified trying to think of another one as I've been going through this. I should have pre-planned this better. Okay. I'm going to say it though, because I feel like it's going to be helpful. So tuboinfundibular. For some reason, to me, that makes me think of like Austin Powers, the like she-bots. And because they're like, oh my God, this is so inappropriate. I can't believe I'm saying it. Because they're like turbo and they're like fun and like, I don't know. I'm like trying to mellow it down a little. But anyways, that's how I remember this one. So tuboinfundibular, because I think of those Austin Power robot girls that are like tubular and fun and like they have, you know, big, yeah, you know, chest area. That's how I remember the side effects for this one. It is increasing prolactin levels that lead to hyperprolactemia. So we're going to see things like gynecomastia or galactorrhea. And then, you know, we might see things like sexual dysfunction. So that is how I remember those. I hope that has. So then when we think of mesolimbic, it's like, okay, that's the one that's kind of left. And that's the one that has to do with too much dopamine. It leads to positive psychotic symptoms. But I mean, of course, right? Just again, like I said, write out that word mesolimbic. Like, what do you think of? So to me, I kind of think of limbs. You know, if we hold out our limbs, it makes a plus sign. So that is increased dopamine equals increased psychotic symptoms. So, I mean, I just did that one off the top of my head, but that's kind of how we can remember these things. All right. So now let's talk about assessment, neuroimaging, and diagnostic procedures. So one thing you may see on the exam is the clock drawing test. This is a simple tool that is used to screen people for signs of neurological problems such as Alzheimer's and other dementias. It's a very easy and quick way to screen a person for dementia. It only requires a few minutes, and impairment on the clock drawing test can be associated with damage to the right parietal lobe. Star. Remember that, right? Parietal lobe is the clock drawing test. So we can see here, a healthy person draws a clock. That's how it looks. Then a person with Alzheimer's draws a clock. That's how it looks. and somebody with Parkinson's draws a clock, and that's how it looks. So we can see the difference. And if we look at this other one, we can see how it slightly changes between early Alzheimer's disease and then late Alzheimer's disease. They really cannot, get that clock correct. All right. We can also use computed tomography, so a CT scan. This provides a three-dimensional view of the brain that differentiates between structures based on density. It provides suggestive evidence of a brain-based problem, but there is no specific testing for psychiatric disorders. These are good because they're widely available and they're inexpensive, but there really is a lack of sensitivity. You can't differentiate white from gray matter. You can't view structures close to bone tissue. There's an underestimation of brain atrophy and the inability to image different views. Then we have an MRI. This provides a two-dimensional image that represents the brain. The advantage is that you can view brain structures close to the skull and can separate white and gray matter. Easily available. It's really superior to a CT scan. Disadvantages are it's expensive. There's many contraindications. Patients might be claustrophobic. The machine looks like a tube, so it can really stress people out. Then we have functional imaging. So this is a technique used to measure the function of areas of the brain, due to the blood flow. It looks at like the blood flow to figure out where the more active parts are. You can use radioactive pharmaceuticals to cross the blood-brain barrier, and this is mainly used for research. These types of tests include EEG and evoked potential testing. This is the least expensive test that conveys information of electrical functions of the CNS. There is a magnetic encephalography, which is similar to an EEG but detects different electrical activity. It's often used in complementary with an EEG. There's single photon emission computed tomography or a SPECT scan that provides information about cerebral blood flow, but it's expensive and there's limited availability. Then there's a PET scan that provides images of the brain when these radionucleotide interact with an electron. It's an expensive procedure that requires extensive resources and a support team. The newest imaging attempts to examine structures in conjunction with function, mainly used for research. So these can be a functional MRI, a three-dimensional event-related functional MRI, fluorine magnetic spectrography, and dopamine D2 receptor binding. All right, and then let's talk about genomics. So for the exam, you do need to understand family history, family trees, and pedigree. You need to understand the pedigree symbols. I mean, don't get too crazy about these, but you should know like a male is a square, female is a circle. You should know what twins look like. You should know the structure of it, how to show like a mother, father, child. It's tools in determining the likelihood of a genetic disorder in a family through inherited patterns and risk of reoccurrence. Pedigree symbols and drawing of a family tree indicate all of these different things. We can see over on the right. Family history starts with the current family, moves back to the grandparents. Autosomal dominant conditions may be present in more than one generation in up to 50% of offspring when one is affected, whereas recessive conditions appear only in one generation, affecting people who have two copies of the faulty gene, one from each parent. And X-linked disorders are caused by faulty genes on an X chromosome. There's genetic counseling, which is a communication process used when a client has a genetic risk and often involves offering a test that could provide information about the genetic status of the person and the possible implication to the family. It's someone whose primary role is to offer information and support to people concerned about an illness that may have a genetic basis. And a referral to a genetic counselor may be needed when a client is anticipating a pregnancy or concerned for health of the fetus. Some genetic terms to know. So chromosomes are structures of DNA in the nucleus of the cell. There are normally 46 in total in humans. DNA is made up of two twisted paired strands composed of sugars linked by 4 nucleotide bases, which is adenine, thionine, cytosine, and guanine, specifying the amino acids that make the proteins. Genes are a sequence of DNA that cause human characteristics to be passed on to the next generation. Genes direct the production of proteins. Messenger RNAs code for amino acids. A phenotype is an observable characteristic of a specific trait and in connection to the genetic contributions to that trait. Gene therapy involves replacing a faulty copy of a gene with a healthy copy of the same gene, and personalized medicine is healthcare based on genetic variability. So studies of population genetics. So family studies investigate the occurrence of disorders in first-degree relatives and second-degree relatives. Twin studies survey the concordant rate of disorders in monozygotes, so identical or dizygotic twins, which are fraternal. Adoption studies investigate the risk of a disorder developing in a child raised in a different environment, but from the same biological parents. A strong genetic contributions have been found for most psychiatric conditions, with a range of 40 to 90% hereditable for some disorders. Genes are risk factors that make a person vulnerable to develop the illness when combined with certain environmental risk factors that increase susceptibility of developing the disorder. Environmental risk factors include prenatal insults, stress, infections, poor nutrition, exposure to toxins, catastrophic loss, and physical and sexual abuse. Most diseases are multifactorial, caused by both environmental and genetic factors. Single gene disorders are rare. Gene expression in disease. So single nucleotide polymorphisms, or SNPs, detect single base changes in DNA sequences. Reducing the penetrance of a gene decreases changes of disease in the person at a genetic risk, and variable expression of a gene for a disorder occurs at the cellular level. There is something called pharmacogenomics, where genes account for differences in the ways that the drugs or enzymes metabolize drugs. Medications may act differently based on how genes affect metabolism. This is why medications work for some people and not for others. Genetic testing or profiling helps identify the presence of these gene variants that may help determine dosing of medication of a gene. So testing for the presence of HLA-B1502 allele, an inherited variant of the HLA-B gene, is required by the FDA for people of Asian descent prior to prescribing the anticonvulsant carbamazepine due to the risk of Stevens-Johnson syndrome. So this is something that you do need to know. You may be asked about this specific gene related to Asian people and carbamazepine. All right, and that is the end. So thank you for watching this, and I will see you on the next one. Bye. Hope this helps.